Hi, I'm John Philpott, and I'm a recent graduate of Carrie Parch's lab at UC Santa Cruz. And in the Parch lab, we study circadian rhythms. And in particular, we are a structural biology lab, so we use biophysical techniques such as X-ray crystallography and NMR spectroscopy in order to interrogate the structure and function of circadian-related proteins. And so to provide a bit of background on circadian rhythms, these are roughly 24-hour rhythms in behavior and physiology that are synchronized to the external day-night cycle. And this allows an organism to anticipate regular environmental changes and to coordinate their behavior and physiology in such a way as to maximally use their energy. And you can see this really well with the sunflower plant, which sort of goes to sleep as the sun goes down. But even before the sun comes up on the next day, it pre-orients its leaves to face that incoming sunlight. Now, much like plants, mammals have circadian rhythms, and fundamentally these are driven by a set of core clock proteins, which I'm showing you here, that you can think of as gears of a clock. And the way that these proteins interact with each other and carry out their basic functions is what gives rise to roughly 24-hour timing. And in particular, my research has focused on the interaction between these two proteins, PER2, or period, and CK1, or casein kinase 1. And so the way the molecular clock works is it's based on a transcription translation feedback loop where you have these transcription factors which bind to DNA and lead to the expression of clock control genes, including their own repressors, PER and CRY, or periods and cryptochromes, which meet up with the kinase in the cytoplasm and eventually translocate back into the nucleus where they shut down their own transcription, making this a negative feedback loop. And this whole loop takes about 24 hours to complete, hence our 24-hour timing. And in particular, my research has focused on the structural aspects of the PER protein. And so we ask questions such as, how does PER recognize other PER molecules and dimerize with itself? How does it interact with the kinase, and how does the kinase phosphorylate regions on PER that either stabilize or destabilize PER? And how does it interact with the other repressor, that cryptochrome, in order to actually repress transcriptional activity? And so these are just some of the questions that my work has focused on. You can definitely check out my poster to get some more detail. And I would just like to thank the organizers for allowing me to be an ARCS scholar and to be able to share my work with the broader audience. And thank you for your time.